sensing our environment and how this perception can affect how we allocate resources. So for instance, you may all know that we keep our body temperature constant, right? And in order to do that, we have to integrate a lot of information. For instance, we have to integrate the temperature of our environment, our feeding status, and all kinds of things. So my lab is very interested in how this mechanism occur and how it can shape our metabolic health and obesity and so on. Okay. All right, so I'm going to break down the talk in 3 parts. One is going to be a short introduction on the sensory adjustment of homeostasis. I'll then talk about a particular neuropeptide that's involved in the communication between sensory information and metabolic homeostasis. And finally, I'll discuss about a neural circuit that can modulate adipose thermogenesis. So this whole idea that sensory perception can actually adjust metabolism came from studies that were pioneered by Cynthia Kenyon in the late 90s. And those were acquired in C. elegans, which is a worm, a nematode. And basically what the Kenyon lab found back in the days was that no matter what kind of mutation you would do that would literally remove the cilia of the worm, so those are the cilia here, the sensory cilia that the worm has to sense its environment, you would get a massive extension in lifespan. So basically, those are lifespan curves, if you've never seen those in C. elegans, and you can see that in black, this is the wild type, and in red, this is the mutation. And for example, here in this particular mutation, you remove all of the sensory cilia and you get a massive lifespan extension. So this really was telling us that neuronal perception of the environment in the worm could influence the rate of aging, and later on it was found it could influence also the rate of metabolism. And when I started to work on this, I also debuted in C. elegans. And this was during my postdoc with Andy Dillon at UC Berkeley and the Salk Institute. And we basically looked at sensory transduction in a few sensory neurons. And we found that the TRPV receptors, so those are ion channels that are responsible for detecting sensory information in the nose of the worms, and they're expressed in specific neurons. When we mutated those, we also gained a significant lifespan extension. So the red curve compared to the blue here. So that was quite interesting, but we didn't really care too much about the worm. What we wanted to know was whether this mechanism was also conserved in mammalian system. And we looked at the mammalian ortholog for those TRPVs, and we looked at TRPV1, which is an ion channel that is involved in perception of heat. So for example, when you're burning your fingers and you feel pain, this is because of your TRPV1 receptors. Or when you eat spicy food and you feel this pungent sensation in your mouth, this is also TRPV1. And those are very interesting ion channels. They are primarily located in the dorsal ganglia neurons. So those are very long neurons, the longest neurons in your body, that innervate very densely the epidermis in the skin. And they go back to these ganglia in the spinal cord here, where they later connect to the brain through the spinothalamic tract. So we looked at this ion channel TRPV1 in mice, and we were curious on whether we would also see a lifespan extension. And that's exactly what we saw. And basically, when we mutated TRPV1 in mice, we observed that animals were long-lived, and that was the case in the males and also in the females. So it's quite important to do both sexes in order to make sure that the effect is real, because sometimes you don't get extension in one or the other. And what we found is that not only the animals were long-lived, but they also had this remarkable reduction in the onset of cancer that they got. So basically, you may know that black 6 mice are primarily dying of cancer. So it's a very good model for cancer if you're interested in that. Whereas you obviously know, this crowd obviously know that in humans, that's not necessarily the case. Cardiovascular disease is usually the primary cause of death in humans. But in the mice, what we saw is that about 70% of the mice died of cancer. And in the trivial knockout mice, that number was reduced. We only had 44%. So that was quite remarkable. And it sort of was telling us that the health span of these mice was also increased, not only just their lifespan. So we tried to understand how this was happening, and we looked at many different things. And one of the things that we found that was really, really impressive in these animals is that the rate of their metabolism was preserved when they were losing TRPV1. So basically, on the left, you can see young mice. This is their metabolic rate when we measure the respiratory exchange ratio. And you can see this nice fluctuation between day and night, which basically tell you that the animals are using carbohydrate metabolism here, and when they're awake, and then when they're sleeping, they're primarily using fatty acid metabolism. So you get these nice fluctuations between day and night. And what is well known is that when animals age, they mostly rely on fat metabolism. They no longer are able to switch between substrates. However, in the trivial knockout mice, we really observe that they maintain this useful ability to switch between substrates like that. So the rate of their metabolism was remained young. They also had improved glucose tolerance throughout life. And we observed that upon aging, their beta cell mass, their pancreatic beta cell mass was higher than controls. And they also had the ability to secrete more insulin upon glucose stimulation. So this was really interesting. We wanted to know what was the mechanism, whether it could be a secreted factor, whether there was a neural circuit responsible for that. And obviously, this potential pathway could be interesting in the context of obesity and diabetes. And this is sort of where I left things, and I started my own lab. So we found something during those days that was quite interesting. It was a neuropeptide, which is called calcitonin gene-related peptide. And this neuropeptide is well known to be secreted by these TRPV1 sensory neurons. So for instance, when you stimulate the TRPV1 receptors, you get not only a response that travels back, an afferent response that travels back to the spinal cord, but you can also get secretion of the CGRP peptide at the periphery of the nerves. So through branching, it's called the axon reflex, you get this release of CGRP. And CGRP goes in a number of places. I will talk about that in the coming slides. And what we found in the mice upon aging is that in the wild type, the levels of CGRP increase with age, but in the trivial knockouts, that wasn't the case. The levels remain pretty low. So we thought that was quite interesting, and perhaps this could be explaining some of the beneficial effect that we saw on metabolism. So CGRP is actually quite well known when it comes to cardiovascular disease. So it has two different forms. So it has the alpha form and the beta form. They're both 37 amino acids. They only differ by 3 amino acids, but they have pretty much the same biological activity. Nobody's ever reported any differences between both. And what they have in common is that they are both really potent vasodilators. So CGRP can be released by the sensory nerves, and again, it's released at the terminal of the nerves, and it can go into circulation where it increases blood flow. Mostly in the microvasculature, this is where it's been found to have the highest effect. And when it comes to systemic effect of CGRP release within circulation, it can also affect the heart, the kidney, and the aorta. It's been found to be protective against hypertension, hypertrophy, and inflammation. So I think in the case of ischemic injury, it's also beneficial. So one thing that is important to say about this peptide is that it's very complex. It has a lot of different biological activity in addition to this vasodilator function. And it not only comes from nerves, the sensory nerves, but it's also been found to be secreted by immune cells and some other cell types. So it's not a very specific sensory peptide, but it's strongly secreted by the sensory neurons as well. So we were interested in this sensory peptide in the context of age-dependent metabolic decline, and we designed this experiment where we acquired old mice from the National Institute of Aging. Those are old wild-type mice. We implanted them with an osmotic pump containing CGRP 837. So this is a peptide antagonist of the CGRP receptor. And basically, after 6 weeks of pump implantation, we looked at metabolic function in those aged mice. And we saw that there was significant improvement in their metabolic rate compared to controls. So that was quite exciting to us and really led the way to try to understand more about this peptide in the context of metabolic disease. So regarding metabolic disease, what we started to do in my lab was to look at type 2 diabetes. And to do that, we use some of the monogenic diabetic models, such as the DB mouse, which you probably know of. So this mouse is extremely diabetic from early age. And it also becomes hyperphagic, so eats a lot, it's obese, and presents many of the features of diabetes, which is hyperglycemia. But it doesn't present a loss of the pancreatic beta cells, so it actually does not lose its pancreatic beta cells compared to other forms of type 2 diabetes. So that's something that's quite important to say when we talk about those mice. And what we found is that in the blood of these mice, the serum level of CGRP were also higher compared to control. And then we look at gene expression of the receptors for CGRP. So there is the RAMP1 subunit and the calcitonin receptor subunit. And we found that in a number of tissues, such as gonadal fat, pancreatic islet, or liver, there was increased gene expression of the receptors, which was potentially leading to some mechanistic understanding of this peptide. So one thing that's I was mentioning earlier, this peptide is very complex. It has a lot of different biological activity. And one of them is that when it's injected in the brain, it will suppress food intake pretty dramatically. So you can see here it's injected in the paraventricular nucleus of the hypothalamus. And basically, gradual increase in the concentration of CGRP lead to a suppression of feeding in mice that are fasted, which is really interesting. And this was done by Richard Palmiter's lab, where they basically use optogenetic to stimulate these neurons in the brain. And they found that upon stimulation of these neurons, you get immediate stopping of feeding in the mice. What was also quite remarkable is that if you delete CGRP in the whole body now, and not just in the brain, what happens is that the increased feeding is there. So that makes sense, because if you inject CGRP, it suppresses feeding. So if you remove it, you get an increase in feeding. But at the same time, the mice had increased energy and metabolism, and they were resistant to diet-induced obesity, which is really counterintuitive, because you would expect that if they eat more, they get fat. So that really was telling us that perhaps the central and the peripheral action of this peptide were quite different. And in order to study this, we had to be very specific about one or the other. So that's what we tried to do. And we used a monoclonal antibody, which does not penetrate the blood-brain barrier, basically. And this monoclonal antibody has been developed by Renat, and it basically is now FDA approved for the treatment of migraine, because this is one of the primary actions of CGRP, which is a vasodilator, and it's supposed to be extremely involved in the in migraine, basically in the pain that is associated with migraine. So this antibody treatment is able to block the circulating form of CGRP really well. That's been characterized before. So we acquired this antibody from this company, and basically we injected that just the way that they did, which is weekly injection in order to maintain antibody level in the blood. And we observed the effect on a number of diabetic mouse strains. So in the DBDB mice, what we observe is that chronic delivery of this antibody led to a small weight loss in the mice. It didn't alter food intake, which is great because that was really important to us, showing that we were strictly working in the periphery. We're not going into the brain with this antibody. And the really, really interesting thing that happened when we delivered this antibody was that it really helped with the hyperglycemia of these diabetic mice. So you can see that the orange curve here are the mice which receive the antibodies. They have a strong improvement in glycemic levels despite harboring diabetes. And they also had an improvement in insulin tolerance, which was shown here. And when we looked at histology, we observed that basically in the different adipose tissue, we didn't see a lot of changes. We saw that there was reduced inflammation in the gonadal fat, which is pretty typical in diabetes. So the mice that got CGRP didn't get adipocytes that were as big as in the control, and they seem to have a reduced number of inflammatory molecules. Macrophages, for instance. They also had improvement in their hepatic steatosis, which is a strong feature of diabetes. And we quantified liver triglyceride. They were also slightly reduced, and as well as serum free fatty acids. So that was very good and very encouraging. We also looked at OB mouse, which is another model, monogenic model of diabetes. So this one is mutation in leptin. So DBDB is a mutation in leptin receptor. And this mouse really becomes extremely obese first, and then also gets diabetes. So it's pretty similar to the DBDB in terms of how they undergo hyperglycemia. It happens a little later, I think, but they really are extremely hyperphagic and also diabetic. So you can see that weight-wise, we saw a very minor reduction in weight in these mice. That was also due to reduced fat mass, and also an improvement in glucose tolerance in these mice. That was great. So the question is, how does it work? What is the mechanism behind CGRP inhibition and diabetic hyperglycemia? So we looked at glucose-stimulated insulin secretion in these mice, and what we saw is that when we give this antibody treatment, we improve GSIS in the mice. So you inject the mice with glucose, and then you measure how much insulin is being released. And that's a very important thing to do in order to understand whether the beta cells are functional. And we saw that in these DBDB mice, insulin secretion was improved. So we then did immunohistochemistry to really understand what was going on in the beta cells in these mice. And you can see in this picture that a few nerves in green are actually CGRP nerves that are going to the islet. You can see like blood vessels here. You can see a pancreatic islet. And it turns out that there is pretty much no immunohistochemistry for CGRP coming from the nerve. So this is the reporter line here. We see like a few discrete cells sometimes, like in this case. But most of the immunofluorescence that's positive for CGRP is found throughout the pancreatic islets. So this is telling us that the reporter model, which is encoded by the gene expression of CalK, is not expressed in the beta cells, but the CGRP immunoreactivity is coming from most likely secreted CGRP that's uptaken by those beta cells. So here you can see another CGRP nerve. And this is a corner of a pancreatic islet. And they're both pretty strongly positive for CGRP. And quite remarkably, when we stain for insulin in this pancreatic islet, we see that there is an overlap in immunofluorescence for insulin and CGRP. So CGRP is not made in the beta cells, but it's uptaken by the beta cells and it colocalizes with insulin. So we then asked what CGRP was doing to these islets, and basically we use a protocol so that was developed by Harshita in my lab. So she collects pancreatic islets, she can culture them for a number of days, and then she can do either RNA extraction or primary stimulation of glucose-stimulated insulin secretion. So when performing glucose-stimulated insulin secretion, what she saw is that if you take wild-type islets, you give them glucose, and then you will see a strong release of insulin as expected. However, if you pre-treat these islets with CGRP, that blocks insulin secretion from the islet. So CGRP is a potent inhibitor of insulin secretion in pancreatic islet. This is what we found. And CGRP also, at the RNA level, blocked the transcription of the insulin gene. And that's pretty much the only effect that we saw. We did RNA-seq, we didn't see anything else besides repression of the insulin gene. And here, this is an experiment in insulinoma cells. So this is a cell line that people use to sort of mimic primary islets. It's very convenient. And here we tried a number of CGRP peptides. So we tried the alpha and the beta peptide, different concentration. And every time we see that CGRP inhibits strongly insulin secretion from the beta cells. So mechanistically, we also looked at other models. We looked at a model of type 1 diabetes, which is the STZ model. And this is basically, you take a wild-type mice, you inject 6 times with streptozotocin, which is a chemotherapy agent that's known to be extremely toxic to pancreatic beta cells. And it leads to hyperglycemia in mice really rapidly. So in those mice, we also gave this antibody, and here we saw no effect on rescuing glucose tolerance or insulin sensitivity, which is really suggesting that in order to do that, we need the beta cells. So this peptide is acting on the beta cells. We also looked at high-fat diet mice, and this is when things got a bit more complicated. So we typically take animals either on normal chow or high-fat diet, and we both gave them this antibody treatment. What we observed is that we also had small weight loss in the mice that got high-fat diet and received the antibody, just like we saw in the DBDB and other diabetic models. And this was due to reduction in fat mass. But here the surprise was that we saw absolutely no effect on improving glucose tolerance in these mice. And we looked several times, repeated that, and we didn't see anything. So the effect that we saw was very, very specific to the DBDB or OBOB models and not in high-fat diet. So I think we're still not quite sure what's happening there. But one of the hypotheses that we have is that in the high-fat diet mice, there is a very rapid expansion of the beta cell pool, which sort of blunts the effect of this peptide. And that's not the case in the DBDB, which do not get this rapid expansion of the beta cell pool. They sort of have a pretty stable, at least for the time duration of our experiment, they have a stable number of beta cells. They don't lose beta cells, they don't gain beta cells in our experiment, because our experiment is only a few weeks. Whereas in this high-fat diet experiment, in the 14 weeks that we do that, there is a very rapid expansion in the beta cell, which we think is blunting the effects of this peptide on glucose, which requires the beta cells, basically. So interestingly, in these mice, when we put them on high-fat diet and we saw this weight loss, but no effect on glucose, we actually see a very remarkable improvement of oxygen consumption in these mice, which is telling us that this peptide may have a dual effect on also on potentially fatty acid metabolism. And it's something that we looked at, and we observed that gene expression for fatty acid oxidation molecules were actually increased, as well as ex vivo lipolysis. So when we collect adipocyte from the mice and we measure glycerol release, which is a measure of doing ex vivo lipolysis, basically, we saw that it was actually improved in those animals when we subject them to isoproterenol, which is a typical treatment in order to induce lipolysis in adipocytes. And we look further into that using seahorse bioscience, which is mitochondrial respiration, where you can do a palmitate oxidation stress test. And what we saw is that when we take primary adipocytes and we stimulate them with CGRP, CGRP actually blocks the ability of the adipocyte to use palmitate as a substrate. So it really blocks fatty acid oxidation. So to sort of summarize this part, we found that CGRP levels are elevated in type 2 diabetes mice, and that therapeutic inhibition of this peptide from the circulation is beneficial in the DB and OB mice, but has no effect on high-fat diet and STZ mice. Mechanistically, we think CGRP has a dual action. It inhibits pancreatic beta cells, glucose-stimulated insulin secretion, and adipose lipolysis. And this is really interesting because the monoclonal CGRP antibodies have been recently approved for the treatment of migraine. And one of them is fremanezumab, I always get it wrong. And this one is from Teva, and actually is this antibody that we are using. So one of the really cool things about this migraine treatment is that people only receive injection every 6 to 8 weeks. So the antibody is very stable in blood, and we think this is interesting because it could be may have potential benefits as well on top of migraine for diabetes, but this remains to be investigated in humans, which we haven't done. So I was telling you before that CGRP is secreted by the neurons, but it can also come from other cell types, such as immune cells. So we saw this really strong effect on knocking out TRPV1 and metabolism, but we don't know if it's entirely due to the sensory neurons arising from the dorsal ganglia or not. So in order to really understand that, we took a different approach. And in this approach, we decided to remove the CGRP neurons this time, very specifically from the dorsal ganglia. So to do so, we use a strategy which is Cre recombinase driven. So it's this advilin Cre, which is predominantly expressed in sensory DRG neurons, and we crossed it with this mouse line, which where GFP is driven by the CGRP alpha promoter. So this is actually the reporter mouse that we use in the immunohistochemistry experiment that I showed. And it also drives the DTR cassette, which is the diphtherotoxin receptor, which allows for ablating the neurons. And basically, upon recombination, you fluxed out the GFP and you get CGRP dependent expression of diphtherotoxin receptor. So you can inject diphtherotoxin and specifically ablate these neurons. So it was important to do a dual strategy with both the CGRP alpha promoter and the advilin promoter because, like I said, CGRP is not necessarily exclusively in sensory neurons. So here we really restricted the expression to the sensory neurons. So having generated these mice, we could finally address what are these neurons really doing in the context of obesity and diabetes. So we subjected the mice to diet-induced obesity and used this diet that is 60% fat, which really leads to a rapid weight gain. So you can see in blue, the wild type are gaining a lot of weight. And what we observed is that the mice that didn't have the CGRP neurons were actually quite lean on high-fat diet. And those here, the squares are the mice on normal chow, which are the controls. So you can see that those CGRP neuronally ablated mice are very lean. And we also looked at adiposity. We found that they had less fat, but they had similar amount of lean mass. They had a strong improvement in glucose tolerance and insulin sensitivity. And I'm not showing here, but we also looked at females, and it was also working really well in females. So we found that those mice also had improved oxygen consumption, which is normal because they are much leaner on high-fat diet. And that translates into increased energy expenditure, both in the light cycle and the dark cycle. This is just another way to look at that, just a calculation. So histologically, we looked at the adipose tissue again, and we found that in the white wild-type mice, you get the brown fat here, you get lipid vacuoles, but you don't really get those in the CGRP ablated mice. And mitochondrial respiration of the primary brown fat, which is we just take biopsies and we put them in the seahorse, shows that no real effect on normal CHO between wild type and knockout. And again, we don't have any weight difference, so that's probably normal. But on high-fat diets, we observe that there is increased mitochondrial respiration in the ablated compared to the normal chow. So really showing that there is more mitochondrial respiration in the brown fat, more utilization of lipids because there is reduced lipid content. And we also measure the temperature of the brown fat, which is something that we typically do in the field to see if there is increased brown fat activity. And you can see that in this circadian experiment, you can see that the mice that have lost those neurons are actually have higher temperature of the brown fat. And we do that at ambient temperature, but also thermoneutrality, which is something that's typically done to remove the cold stress of the lab environment. So now I have to give you a bit of a very brief introduction on thermogenesis. So adaptive thermogenesis is obviously the process of generating heat in organisms, and it depends on these various parameters, which in a mouse is obviously its basal metabolic rate, its physical activity, the thermic effect of food. So if you give high-fat diet, this increases energy expenditure. It's well known that food has a thermic effect, so it's called diet-induced thermogenesis, and also the temperature of the animal. Those are like all the parameters that are important to take into account so that the mouse can maintain, again, its temperature at a constant level. And this is done by two different organs. So the muscle can perform shivering thermogenesis, so cold induced, but that's not really sustainable. This is something, for example, if you're cold on the beach, you're going to put it, you're going to have goosebumps and shiver, but that's not really something sustainable. You want to have a more sustainable way to maintain your temperature. So one of them is diet-induced thermogenesis. This is also dependent on brown fat, or cold-induced non-shivering thermogenesis, which is what a lot of us study in the context of obesity, because this depends on the brown fat capacity to really use all the lipids and stores in your body in order to really bring up that temperature to the required level. And this phenomenon is extremely important. It's really obviously extremely controlled. So when I say that, it means that the brain is involved, obviously. So if the body is subjected to cold, this is sensed by the skin, the brain, or in the case of the internal temperature, by the viscera. And this is integrated at the level of the hypothalamus, which will lead to counter-regulatory measures in order to adjust things. So one of these is the shivering thermogenesis that I was mentioning, and one of them is this adipose thermogenesis and vasoconstriction. And this is typically driven by sympathetic nerves within the body in order to really bring body temperature to a constant level. And those sympathetic nerves are extremely abundant in adipose tissue. You can see here, this is the subcutaneous fat, and those are data from Paul Cohen's lab, where basically you have a crazy innervation of adipose tissue in the subcutaneous fat. So it's densely packed with these tyrosine hydroxylase nerves. And the adipocytes, they contain beta-adrenergic receptors. So the sympathetic nerves can release catecholamines, such as noradrenaline, for instance, which will act on these beta-adrenergic receptors. So in the context of white fat, it leads to lipolysis. In the context of brown fat, it leads to mitochondrial respiration and uncoupling to use these fatty acids in order to generate pure heat. So instead of making ATP, the mitochondria are going to make heat out of those lipids. And it also happens in those beige cells, which are contained in the subcutaneous fat and have the ability to become more like brown-like cells. So we looked into whether those mice had increased adaptive thermogenesis. And we found that when we stimulate them with a beta-3 adrenergic molecule, which is CL31623, it's something that people in the field typically use to stimulate this system, we found that in normal chow, CL lead to a transient increase in oxygen consumption, which is usually occurs within 3 hours of injection. And it happens in the wild type, but it also happens in the CGRP ablated and actually is more robust in those mice. And when we looked at oxygen consumption in the mice where the CGRP neurons were ablated on high-fat diet, and this is only 2 weeks of high-fat diet, so there is not yet a weight difference among the groups, you can see that oxygen consumption is already higher in those mice, both in the dark cycle and the light cycle. However, when we inject CL, we also see a robust increase in VO2. But in that case, it's only a minor transient increase, which is sort of telling us that most likely there is increased beta-3 adrenergic signaling in our mice, but it's not big difference on high-fat diet. It's a bigger difference on normal chow. So because we see this increased oxygen consumption in those mice and this weight loss, we investigated whether they had increased white adipose tissue lipolysis. And we found that the gonadal fat pads showed smaller adipocytes in those mice. And they actually had, when we tested it, ex vivo lipolysis, they had increased ex vivo lipolysis compared to the control. So those are basically vehicle-treated adipocytes here. There's no difference, but when we subject them to isoproterenol, which is again this molecule that stimulates lipolysis quite well through adrenergic signaling, you can see that in the CGRP ablated mice, we gain quite a lot of glycerol release, which is ex vivo lipolysis. And gene expression showed that multiple genes involving free fatty acid oxidation transcriptionally were increased in our mice. So if you look at vehicle-treated wild type versus vehicle-treated CGRP knockout, that's the case. When we inject the CL, we get an increase in all of that because CL dramatically increases lipolysis and gene expression of lipolytic genes, but it's sort of blunted between the wild type and the knockout, which is again telling us that it's not necessarily what's going on in our mice. And remarkably, the CGRP ablated mice have increased gene expression of ADRB3, which is encoding for the beta-3 adrenergic receptors in white fat. So we think what is happening in those mice is that they most likely have increased beta-3 adrenergic signaling in white adipose tissue that leads to increased lipolysis and weight loss, and therefore utilization of those lipids by the brown fat mitochondria. But these mice do not show any browning, so the subcutaneous white fat does not show increased UCP1. I'm going to go quickly through that. And the brown fat also doesn't show increased UCP1, which is really to tell you that the beta-3 signaling that we see, the gain of function that we see, we think is really specific to the white fat and not the subcutaneous fat or the brown fat. So why are those mice losing weight and showing increased lipolysis on high-fat diet? So we think the explanation is that they feel colder at ambient temperatures. And we measure that using infrared imaging of the tail. So you can see, so this is the tail, the base of the tail, you can see here. And when you can collect the temperature reading here by infrared thermometry, it tells you about basically vasodilation, vasoconstriction based on the temperature of the tail. And you can see that when you look at it on a circadian manner, those CGRP ablated mice always feel colder than their control. And that's the case on normal chow, that's the case on high-fat diet. When we bring them to thermoneutrality, we do see an improvement, so they feel less cold. But even on high-fat diet, they still show colder tails at thermoneutrality that we saw at the peak of the night. Remarkably, they feel more cold, but they actually are not cold. So they have counter-regulatory measures to bring up their core temperature in order for it not to drop. So this is just showing you normal chow mice. You can see that the core temperature is the same. And this is subjected to a cold stress test. So you would put the mice at 4 degrees for 5 hours. And you can see that their core temperature doesn't drop. So they found a way to counterbalance the feeling of cold. And others have shown that if you ablate the TRPV1 neurons, basically, and you inject insulin, which is a menthol-like molecule, so it gives a very strong sensation of cold, the mice actually are going to feel more cold. So there is more electrical activity of the neurons when they sense insulin. So basically the way it works is that you get these TRPM8 neurons that are called sensing neurons in the DRG, the dorsal ganglia. And the TRPV1 neurons are presumably inhibiting these TRPM8 neurons. So if you remove these TRPV1 neurons, you get a gain of function in cold perception. And so we asked whether that was the case when we removed those CGRP neurons. So we injected insulin in the mice. And what we observed is that insulin injection leads to a very strong perception of being cold. And the tail temperature drops dramatically in both genotypes. But remarkably, mostly in the CGRP ablated mice, the brown fat temperature increases, which really shows that they feel more cold than the wild type. So they have a gain of TRPM8 activity. And that's really interesting because other people have studied loss of TRPM8 activity in the context of obesity and high-fat diet. And they found the converse effect that we found. So if you lose TRPM8, if you lose the ability to sense cold, the mice actually become obese, hypometabolic, and actually hyperphagic because they have the need to eat constantly, I think. So this is really cool. So we asked whether the CGRP neurons were required in the brown adipose tissue because they're predominantly contained in the skin, but we also found that they're expressed in the brown adipose tissue. So we collaborated with Paul Cohen at the Rockefeller University to look at brown fat, and we found some discrete population of CGRP neurons there. Excuse me. And basically removing those neurons through specific diphtheria toxin delivery in this tissue had no impact. So we think really those neurons are required in the skin to have these effects. So I'm not going to go too much into details, but basically the mice have no phenotype here. So basically, overall, there is a lot of interest in obesity research as to using adaptive thermogenesis in a way to treat obesity. And this is an experiment that was done many years ago, where basically individuals were subjected to a 14-degree cooling protocol every day. And this is a lean thermoneutral individual. And when it's cold exposed, you can see 18-deoxyglucose PET utilization by the brown fat. It really shows that it's activated brown fat. If you do that in overweight individual, what this study and actually other studies have found is that you get a little bit of brown fat activity in some of these obese individuals, which is really promising because it could really help obese individual manage weight gain, basically. So the model that we are working on is basically the following. So temperature is sensed, outside temperature and environmental condition are sensed in the skin by these DRG neurons that express TRPV1 or TRPM8. And basically those will inform the brain about the external temperature, and that will lead to counter-regulatory measures. That's not something we have investigated in the present study. To either use vasoconstriction or adipose thermogenesis in order to maintain core temperature. And in our case, what we found is that upon loss of the CGRP neurons, there is increased perception of cold, which leads to increased fuel utilization by the brown fat, increased adipose tissue lipolysis, and also reduce CGRP secretion in order to lead to vasoconstriction and to conserve heat, basically. So in the first model, I showed you that if we remove CGRP, we get beneficial effects, and CGRP is a potent vasodilator. So we think that upon warm temperature, there is more CGRP that is released, but upon cold temperature, CGRP is decreased in order to induce vasoconstriction and conservation of heat, basically. So we think that this is really interesting, that potentially this could be utilized for better management of obesity and diet-induced thermogenesis, really. So one thing that's important to think about is that some of the manipulation of this pathway can lead to hypothermia. We haven't seen that in our mouse model, but this is really important. So in the future, really understanding how uncoupling hypothermia and weight loss works can have a very strong therapeutic potential. So I want to thank everyone that worked on this project, and there's quite a lot of people. John, Mandy, Alex, and Harshita have worked on the peptide treatment in the first study. Kuldeep, Harshita, Nancy, Peter, Peja, Yidan, and Edward, which I forgot to put here. He's here, he's now in my lab. I've worked on the neuronal innervation study. And I want to thank our collaborator at Rockefeller, Paul Cohen, and Jingy and Will, who have done a lot of adipoclear staining of our mouse samples, as well as the mice line that we use, the antibodies from Tiva Pharmaceuticals, and Stephanie Cora at UCLA that helped us with some reagents. And finally, our funding sources. So we're very fortunate to receive funding from the American Diabetes Association through the Pathway Program. And we also got a Larry Hilbaum Foundation Award to do some of this work, and a pilot award from the Diabetes Research Center of UCSD. Thank you for your attention.
Thank you, Celine, for this great seminar, which opens our eyes to sensory regulation of metabolism in the seminar series. So we can take questions if anybody wants to raise hands. And I can, okay, I'll start with Esther and then I'll ask more. Go ahead.
You can ask first, April.
No, go ahead. I'll be the last.
So, Celine, that was a wonderful presentation. Thank you. And as a vascular biologist, I was wondering how much of your phenotype could be derived from the vasoconstriction?
I think all of it. I think most likely that's the case because the primary function of this peptide is really vasodilation. It would be really interesting to be able to uncouple the vasodilation from the other aspects, but I don't know if we would be able to do that.
If you
But
give
yeah,
them nitric oxide or something, then that
yes, something like that.
Yeah, that would be quite cool. Yeah, but wonderful. It's quite complicated, but I think the brain does a lot of the regulation. So
Yes, I think so too.
Yes, Mabruka, please.
Hi, that's really very, very interesting talk. So I enjoyed every aspect of it. So I was wondering, I'm trying to follow up with a question. So it seems like this peptide is doing something in the brain that's different than peripheral body, right? And you mentioned that in your talk. So I'm trying to link it to the receptors. So how the receptors, is there like a different receptor phenotype in the brain that's different in the body, and that's why you saw the different response? Or do you think that peptide's being taken by different cells, and this is why you see? So in other words, is it cell mediated or is it receptor mediated?
So in our models, we tried really, really hard to focus only on the periphery, and I didn't show it, but in the second model, we showed that we are not ablating any neurons in the brain. In the antibody treatment, we also use this antibody that does not cross the blood-brain barrier, and we never saw changes in food intake, which is really good. Because this is really what this peptide has been shown to do in the brain, is really to change food intake. So one injection of this peptide suppresses food intake. So in our case, we really tried really, really hard to stay away from that because the biological activity is so complicated already. It has many different properties. You saw like pancreatic beta cell, insulin secretion inhibition, adipose tissue, anti-lipolytic effects. It also works in the cardiovascular system and can also modulate immune cells, change TNF alpha. It's a very versatile peptide, which makes it really complicated to study. So we tried as hard as we could to narrow it down to periphery and stay away from the brain. In terms of the receptors, it's even more complicated because it has action on the calcarel and RAMP1 subunit, which work together. This is its primary receptor, but also it's been found to be able to bind to the amylin receptor and the calcitonin receptor. It has less affinity for those, but it still can bind there. So it really is quite complex. And amylin and calcitonin, which are I would say close orthologs of CGRP, also can bind on the CGRP receptor. So it's quite complicated.
That's really interesting. So I'm a clinician by training. So every time when I saw somebody speaking science, I want to see how that's going to help patients. So is there any diabetic patients that you can relate to? You saw literally an increase in the peptides in their serum, or is there any research is being done on this peptide in diabetic patient in general?
So it's not, we haven't done any work on patients, but I think I'd be really interested to move to humans, and especially because the antibody is already FDA approved, which is a huge benefit in our case. We know that can be done. So I'd be really interested in pursuing that. But like, I don't know if it's going to work necessarily because we saw very beneficial effects in the monogenic diabetes model in mice. And it may differ quite a lot from human diabetes. And again, human diabetes is just not one disease, it's a combination of things. People have diabetes for many different reasons. There are genetic factors, there are environmental factors. So it may help some people, but not necessarily all people. So it'd be quite interesting to find if it can do any good to some patients at least.
That's really interesting. Thank you very much.
Thank you.
Adil was next.
Hi, thanks. That was a great talk. I just want to make a quick announcement for the trainees that we will have open networking after the session. And so I'll ask my question during that session, but thank you.
Oh, okay. All right. In that case, Mark is next.
Hi, that was a great talk. I really enjoyed it. Hello. I have a question on this dual action on beta cells and lipolysis, right? So as you know, insulin will repress lipolysis. So in vivo, do you think that the action, the adipocytes might overcome the ability of insulin to suppress lipolysis through an independent pathway, or maybe in vivo, could it be possible that the improvement in GSIS comes by improving adipose tissue function, right? Because in the end, in vivo, you have the crosstalk between the two tissues. So how do you?
That's a good question.
Work out.
Very good question. So we saw effects on primary islets, and we saw effects in vivo. And we are not the first one to describe those in vivo effects. So it's been done. Actually, it was something people used to do a lot in the 80s, look at CGRP and pancreatic insulin secretion.
Mm-hmm.
And it sort of fell out of trend for some reason. But it seems to have a very strong effect on insulin secretion, at least in vivo. In terms of having the dual effects on lipolysis, so yeah, possibly. So it's an inhibitor of insulin secretion. So you could imagine that if you get less insulin, you may indirectly also affect lipolysis, in addition to the direct effect that the peptide may have itself on lipolysis. But like, we haven't really tried to do that in vivo in terms of looking at lipolysis. I guess that's something
maybe related to that could be specifically participating in beiging, right? Because beige adipocytes behave differently than white adipocytes. So maybe it's about having a specific action on the beige adipocytes promoting lipolysis, but not on the white, right? That could also...
Yeah, we've only looked actually in subcutaneous SVF cells. So we've never really done differential analysis of the white versus brown versus beige. So we've always taken SVF isolated from the subcutaneous fat, just because it was easier, I guess. But yeah, it would make a lot of sense to look more into that.
Cool. Thank you, and congrats.
Thanks, Mark. I have two questions that remained in my mind. I had more questions, but I'll catch you and ask you later, Celine. So one of them is in the high-fat diet, where you lost some of the strong regulation on metabolism. Did you check if the inflammatory nature of this high-fat diet altered maybe the entry of this peptide into the pancreatic beta cells? So that's one thing that occurred to me. And maybe that's why, because we can't get in.
That's really interesting. I've never thought about that. So I'm glad you bring it up, because we really did not understand why it really didn't want to work. And, you know, we tried many times.
Yeah, because there is the whole inflammation impact you get with high-fat diet that you don't really get with the other models.
Yeah.
And the other thing that occurred to me is, because I was reading about this recently, is, you know, the focus is on thermogenesis, but have you considered from the brown fat perspective, but maybe there are other peptide messengers being generated to regulate your metabolic phenotype?
Oh, for sure. I'm sure there's many peptides, and there's a battery of peptides that we now know can impact on brown adipose tissue. Brown adipose tissue itself can secrete several cytokines, I guess. So yes, and I mean, obviously, noradrenaline is coming from the sympathetic nerves, is heavily regulated by the brain. So we think that, yes, for sure, this is just a small aspect. But like, what we're trying to say is that this is maybe one of the primary things that the organism utilizes in order to adjust everything else. So really, by sensing the temperature from the skin, the brain starts to form a response that will eventually lead to a whole array of counter-regulatory measures, including adjusting vasodilation, vasoconstriction, and influencing brown adipose tissue, white adipose tissue, and so on. So.
Thank you. So any more questions, or should we just move on to the metabolism trainee session? All right, so then I'm making Mabruka host, and sure. It's all yours, Mabruka, and great seeing everybody.
Thank you so much for the opportunity.
Take you so much.
Thank you very much.
The only thing is,
and you can stay if anyone want to stay behind, so interact. I can see Ada came back. So
I have a question. If Adil will let me ask my question first.
So, I don't know how to stop the YouTube.
So, okay, you can leave us on YouTube.
Okay, if you guys want, I can just restart the session if that's what you prefer.
No, it's okay.
I think it's fine.
Okay.
All right, sorry about that. I have to figure this out. All right.
Thank you.
Yeah, I was wondering again about the difference between the high-fat diet model and the OB-OB or DB-DB models.
Yeah.
Whether maybe the peptide has some effect, maybe downstream of the leptin receptor, and that's why it's working on the DB-DB OB-OB models, but not on the high-fat diet models.
Yeah, so that's a really good point. So one thing that's a bit frustrating, I have to say, about when working on obesity and diabetes in mice compared to other models, so for example rats. So if you give mice high-fat diet, they will never get diabetic. Never. They will get insulin resistant, hyperglycemic, obese, hyperphagic, but they will never have an actual diabetes where they lose beta cells and they get extremely high level of glucose. So that's why people have eventually started to use genetic models, because it's kind of the only way in mice to get to mouse diabetes. So I think this is something that needs to be looked at more into details. A lot of people use the OB and the DB, but those models also are not necessarily the best. So for example, we use the black 6 mice in our case, and if you were to use the OB and the DB mice in a different strain, so I think it's the B6 strain, those have a completely different diabetes, right? So the black 6 and the B6 do not behave the same, even though they have the same DB mutation or OB mutation, for example. So it's a problem. I mean, it really shows that the strain, the genetic background can really change a lot. And when I say a lot, I mean the black 6 DB will have a certain number of beta cells. They will go, I think, a small loss of the beta cells over time, but very small. Whereas the B6 DB will lose their beta cells. So those ones are really behaving quite differently. They get different kind of diabetes. And we tried in those actually as well, and we saw like our peptide antibody treatment was working in the B6 as well. But yeah, I have to say, I think the rats, for example, rat models are better because you can do high-fat diet and the rat will get diabetes. So it's maybe something we may want to do in the future is to sort of try the rat as a model. because the mice, yeah, it's so complicated, and you really may not really mean much sometimes, unless you try all different strains that exist. But that's a lot of work.
I'm gonna follow up with the difference between the physiological difference between mice and early on in your presentation you show very beautiful this wake up and sleep cycle when there's carbohydrate metabolism. That's something I've never known about it, so that's something new to me. So, and then kind of the mice, they lose this when they get older. So as a human being, are we also do the same thing? Do we have more, we rely more on carbohydrate metabolism during sleep? Is there any idea?
Yeah, so mice are nocturnal, so us it's opposite. So basically, yes. So it's been shown that with aging, the flexibility of the metabolism, this is how it's called, sort of declines. And that's why animals and humans tend to get more adiposity over time with age. So you know how, yeah, you know how, like in the case of many humans, you get a little bit more fat over time. And that's definitely due to this metabolic inflexibility. So it really is a mechanism that's been somehow studied, but it's still not fully understood. Where the mitochondria, so the mitochondria, they're able to use two different types of substrates, right? They can use either lipids or they can use carbohydrates. Lipids are great when you're sleeping because you are not uptaking any food, so it's easier to acquire lipids from the adipose tissue, for instance, and those will generate a lot of ATP. Carbohydrates also generate ATP, but they generate a bit less actually. So and one thing that's been shown is like over time you get I think increased phosphorylation of the AKT subunit, no, the IRS1 subunit, which leads to some sort of insulin resistance in the cells and would be associated with this metabolic inflexibility phenomenon where it sort of blocks mitochondrial ability to rely on glucose, basically, when they can no longer uptake glucose the same way, and they sort of switch to using lipids entirely.
And has this peptide restored that?
So how these neurons sort of maintain a more useful metabolism? So we think it's because they sort of trick the brain into perceiving the environment in a way that the brain takes decisions differently and sort of uses resources in a way that's more conservative over time. And also the neurons we found have this effect on insulin secretion in the beta cells. So we really saw, like the mice, when we aged them, that they had a very well conserved beta cell pool. So there is no decline in the beta cell mass over time. So we think the preservation of the beta cell integrity and maybe the effect on, I don't know, adipose lipolysis and potentially how brain integrates signals may contribute to improving how the metabolism remains more useful over time. And we think this is why the mice were long-lived, is really because their metabolism was more useful. We tried to look at many other things that nothing else worked. So yeah.
It's very beautiful.
Yeah, thank you. It would be great to be able to use that in humans in some way.
It is. It will be really cutting edge. I can see that. It's gonna be beautiful. So I'm gonna ask you about career. So I can see your CV is very impressive. So how did you make it there? Is it like and really hard work, really extensive amount of lab work and data? So we are at the stage of transition. So we want to know and learn from your experience. If you don't mind, we can share that.
Yeah, no, of course. So, I mean, I don't know. I don't think there is necessarily a recipe for, you know, for success, or I don't know. I don't think I found it, by the way. I think it's important to be passionate about what you do and keep having fun in doing your work. And I think to me, that's the most important part. I don't really care too much about my career in, you know, an academic way. I just care about what I do, and I want to do it with the people that, you know, bring me joy every day, like people in my lab that I love working with, and so on. So I think this is really the key for me. I've never really made any plans about career or anything else. Just things just happened in my case. I think what I can tell that for sure helped me get to academic position was the fact that I did my postdoc with Andy Dillon, who is somebody that's really well established. He's a HHMI investigator in the US, so that's and obviously the work that we managed to publish was published really well. We had this paper in Cell. I also had another paper in Cell Metabolism a few years after that, and thanks to him, yeah, things really he also showed me that, you know, it's important to just do the research that you love and do it in a fun way. And that was really my driver for everything, I have to say. Like, he pushed me into applying for academic positions. That was not even on my radar back then. He also pushed me into applying to certain funding. So I got this ADA Pathway Award, which I got as a postdoc. And it was great because it's a lot of money, and that's something that I still have. It's a 7-year award. And I never thought I would get it. Like, if I had listened to myself, I would not even have tried. But yeah, so I think I got a lot of really good mentoring from him in that regard. And he helped me go into that direction, because as a woman, I don't really, you know, have necessarily the highest opinion of my performance or anything. So I don't think I can do this most of the time. And he was like, yes, you can do it. You should do it. And I was like, really? Why? So he was very helpful. I also got mentored by great people in the aging field. Anne Brunet, she's one of them. She's a professor at Stanford. She's also one of the few women that I know, that I knew back then, that I saw she was, you know, French like me, very successful. She had her own lab. She's like, yes, you can do this. So you should try. And overall, people were really supportive. And I think one of the advice that I have is to try to surround yourself with people that are mentor figures. And I'm sure you have like professors around you that you talk to, and those people are always happy to help. And sometimes the most difficult part is to ask for help, you know. And this is really like the philosophy that I have now. So I'm, you know, always trying to help younger people, because I was so glad that these people helped me. I don't think, you know, I have any kind of career without them. And sometimes all they did was just to open my eyes, because my eyes were closed to the possibilities, really. So I would say, yes, ask for help, ask for feedback, and create like a network of people that will be happy to, you know, advise you, because you need so much advice all the time in this work.
Very true. So just follow your passion, go with whatever that passion will take you.
I think so. And like, you shouldn't try to do things that don't make sense for you, you know. because science should stay fun. I think to me that's the most important part. It should stay fun and it should stay exciting.
Yeah, it's fun, but it's at the same time it is a bit tough, you know. But
It
yeah, I
is
agree.
tough. You have to make a lot of sacrifices, you know, we all do. Most of the time it's like you don't get to live necessarily where you want to be. It's hard to have it all.
Okay, totally agree. So we are 19 minutes past after the seminar. So guys, if you have any more questions, probably we will leave it there. Thank you very much.
Thank you.
I see Ada.
Oh, I just wanted to say thank you. That was fantastic. And thank you for your advice.
Yeah, and like I'm happy, like seriously, if you really ever, you know, want to talk more about career and need feedback, like I'm happy to do that. Just email me and we can schedule a call.
Thank you so much.
You're welcome. Thank you, guys.